Good morning students. Last class we have discussed about the genetic code. Okay, so DNA is a genetic material. Inside the genetic material it is that genetic information it is coded, okay, in the form of genetic code that we have discussed. So mainly in this replication of DNA, okay, the DNA, one strand of DNA, it is transcribed into the RNA. In the RNA, it is they can produce in three different types of RNAs: mRNA, tRNA, and rRNA. Then that RNA they can synthesis the protein. That method it is known as the translation. In the method of translation, they can synthesis the protein. Okay, this is the replication of DNA. So in this format, DNA they can produce in a fragment of DNA, they can produce in a same multiple copies of the DNA. Then that time they can carry the genetic information. That genetic information through the genes they can transfer from generation after generation. Okay, that is the method. That is the way the genetic information they can transfer from generation after generation. That we have discussed in a last class. Then the last class we have discussed about the salient features of genetic code. Also we have discussed. So mostly 20 different types of amino acids are there. That 20 different types of amino acids it is involving. They can produce 64 possible triplets. From that 64 triplets, 61 codons are coded for the amino acids. Then the remaining codons are stop codons. They cannot involve to synthesis the protein. They can stop the process of translation. Okay, so that fully we have discussed in the last class. What are the salient features of genetic code? That also we have discussed in the last class. Then we have discussed about the mutation. What is the meaning of mutation? Sudden changes. Okay, suddenly changes occur in genes, that is leads to the modifications, that is known as the mutation. So how the mutation it is involving to changes in the genetic code. Okay, that we have discussed. For example, sickle cell anemia, it is a disease, blood disease. In a normal normal blood cells it is present. Normally the blood cell DNA 5' to 3', that is the polarity. Then 5' to 3', how many amino acids are there we have discussed. So in the 6th amino acid, glutamic acid, it is changed into the valine due to the mutation. So normal blood it is changed into the sickle cell blood. Okay, normally red blood cells are biconcave disc shaped structure, that it is changed into the irregular sickle cell red blood cells. That we have discussed everything in the last class. Okay, so how the mutation it is involving, that fully we have seen. Then we have discussed about the Hubble hypothesis. So in this hypothesis the triplets, okay, the first and second position is the constant, third positions are flexible, maybe they can change, okay, in its codon and then in anticodon. That is known as the Hubble hypothesis. That also we have discussed in a last class. So in this class now we are going to discuss about the tRNA, the adapted molecule. So tRNA, transfer RNA. So in the process of transcription, single strand of DNA, they can produce the three different types of RNAs. Okay, messenger RNAs, they can pass the messages from cell to cell. tRNA, they can transfer the information from one cell to another cell. Then ribosomal RNA, it is involving in the protein synthesis. So these three types of RNA, it is synthesizing in the process of transcription. Okay, now we are going to discuss about the tRNA. How this tRNA it is involving in the adapter molecule. Okay, tRNA, the adapter molecule, the transfer RNA or tRNA molecule of a cell acts as a vehicle that picks up the amino acids scattered through the cytoplasm and also reads specific codes of mRNA molecule. So what is the main functions of this transfer RNA? It is acting as a vehicle. What is the uses of the vehicle? It is helpful to transfer from one place to another. So like that, this vehicle, tRNA vehicle, it is doing the work inside the cell. Amino acids are scattered here and there. Okay, this is a cell. So inside the cell amino acids are scattered here and there. So this tRNA, okay, it is acting as a vehicle when it is moving from here to the one place to another. So that time they can pick up the scattered amino acids inside the cytoplasm. And then another one, it is reads to the specific code of mRNA molecules. mRNA synthesis ஆகக்கூடிய அந்த specific code அதுலேயும் இந்த tRNA involve ஆயிருக்கு. So it is known as the adapter molecule. Okay, can you understand? So this term was postulated by the scientist Francis Crick. Francis Crick only given the name tRNA as a adapter molecule. So what is the main functions of this tRNA? tRNA, it is acting as a vehicle, then they can collected the amino acids scattered through the cytoplasm of a cell, and then also it is reads a specific code of a mRNA molecule. So it is known as the adapter molecule. Then the two-dimensional clever leaf model of tRNA was proposed by Robert Coley. The clever leaf model, two-dimensional structure, it is postulated by the scientist Robert Coley. Okay, here they have given this is the modified structure, secondary structure of a clever leaf model of a tRNA, transfer RNA, tRNA structure. Okay, so this actual structure of this tRNA is a compact, it is look like a inverter L-shaped structure, inverter L-shaped structure, this tRNA molecule structure. Then the clever leaf model of tRNA shows the presence of 3 amps. So 1, 2 and 3 amps are present. We can say amps or loop, 3 loops are present, D loop, anticodon loop, and then T loop. Like that 3 loops are present or 3 amps are present. Okay, first one DHU, D loop, and then anticodon loop, and then T loop. So 3 loops are present. Then these loops, then these amps or have a loop such as amino acid binding loop. Here amino acid they can bind one of the loop. That loop it is known as the amino acid binding loop. Then anticodon loop, and then ribosomal binding loop. So like that 3 loops are present. Amino acid binding amino acid binding loop, anticodon loop, and then in a ribosomal binding loop. 3 loops are present. Okay, in there 3 ends loops, 3 loops are present. In addition, it also shows a small loop. That loop is known as variable loop or a extra arm. Okay, variable loop or then in a extra arm. So it is look like a inverter L-shaped structure. Okay, this structure it is proposed by the scientist Francis Crick. Okay, then the two-dimensional clever leaf structure it is postulated by the scientist Robert Coley. Then this clever leaf structure it is look like a inverted L-shaped structure. Okay, inverted L-shaped structure. 3 at their end 3 loops are present. Then that at the end starting amino acid it is acceptor end it is present. That amino acid acceptor end one loop is there, that is known as the amino acid binding loop. Then anticodon loop, then ribosomal binding loop. Okay, here ribosome they can bind at the end. So like that 3 loops are present. Then another then one of the extra or extra loop is present. This is known as a variable loop. Okay, so the amino acid is attached to one end and consists of 3 anticodon nucleotides. So here one end 3 codons are there. Then in opposite 3 anticodons are present. Okay, so the amino acid it is attached to the one end consists of and the one end and consists of 3 anticodon nucleotides. Okay, 3 anticodon. So the anticodon pairs with the codon in mRNA, ensuring that the correct amino acid is incorporated into growing polypeptide chain. So when this codon and then anticodon it is present in a correct pairing in the mRNA, so it is helpful to grow in the length of the polypeptide chain. Okay, it is useful to grow the length of the polypeptide chain. Okay, this is the clever leaf model of a tRNA. Okay, so variable loop. So like that the 4 loops are present. Then the amino acid is attached to one end. That end it is known as the amino acid acceptor end. Amino acid accept in the one end. This end it is known as the amino acid acceptor end. Okay, and the other end consists of 3 anticodon nucleoside. So opposite to this end 3 amino acid anticodon end nucleotides are present. One side amino acid acceptor end, then opposite what is that? Anticodon nucleotide end is present. Okay, anticodons are present. Then the anticodon pairs with a codon in mRNA, ensuring that the correct amino acid is incorporated into a growing polypeptide chain. So when this amino acid, then they can coded with the correct of anticodon. This both the ends are present in a correctly. It is helpful to leads in a growing in a polypeptide chain. அப்படியே polypeptide வந்து கரெக்டா இருக்க அந்த பேஸ் பேர் வர எக்ஸ்ட்ரா length chain உடைய length வந்து increase ஆயிட்டே போகும். So 4 different regions of double stranded RNA are formed during the folding process. So when it is producing in a 4 different regions, amino acid acceptor end, then the T loop, the anticodon loop, D loop, and then in a variable. These regions fully, okay, 4 different regions of double stranded RNA are formed during the folding process. Folding இருக்கப்பதான் இந்த 4 regions வந்து 4 different regions வந்து develop ஆகும் double stranded RNA molecule. Okay, then modified bases or okay, modified bases are especially common in tRNA. So in a tRNA it is present in a modified base pairs are present. Modified bases are especially common in tRNA because already we have seen in this genetic code, that mutation in genetic code we have seen this third position, it is in a for what? Flexible. It is possible to change in its codon and then in a anticodon. Flexibility it is present. So modified bases are especially common in tRNA. So wobbling between anticodon and codon allows some tRNA molecules to read more than one codon. So in the wobbling hypothesis மூலமா இந்த third positionல என்னது flexibility இருக்கிறதுனால maybe codon anticodon எப்படி இருக்கும் change ஆகும். Okay, இப்படி change ஆகிறது தான் என்ன செய்தான், it is helpful to okay in a wobbling between the codon and then in anticodon allows some tRNA molecules to read more than one codon. ஒரு சில tRNA molecule இங்க நிறைய codons வந்து இருக்கும். So the process of addition of amino acid to tRNA is known as amino acylation or charging, and the resultant product is called amino acyl tRNA or otherwise known as charged tRNA. So this according to the wobbling hypothesis, that wobbling it is present in between the codon and anticodon. Okay, this tRNA molecule it's read to more than one codon. Okay, in the process of addition of extra, when the process this wobbling hypothesis it is involving, it is present wobbling it is present in between the coding and then in codon and then in anticodon. Okay, then extra additional amino acids of tRNA, it is amino acids, excess of amino acids it is for binding with the tRNA molecules. Okay, excess of amino acids they can bind with the tRNA molecule. Okay, when the excess amino acids they can bind with this tRNA molecule, that tRNAs are known as the charged tRNA. Okay, the tRNA it is known as charged tRNA. The process of addition of amino acid to tRNA is known as amino acylation or charging. Okay, it is one of the process. In this process, excess of amino acids, additions of amino acids, it is present in the tRNA molecule. Okay, in the tRNA molecule, excess of amino acid they can go and bind. Okay, so this tRNA it is known as the charged tRNA. That process is known as amino acylation or charging. Okay, what is the meaning of amino acylation? Excess of addition of amino acid to the tRNA molecule. Okay, excess addition of amino acids to the tRNA molecule is known as amino acylation or charging. Okay, so result of this process they can produce the charged tRNA. Okay, without amino acylation tRNA is known as uncharged tRNA. So that amino acylation process it is not occur in a tRNA, that is known as uncharged tRNA. Amino acylation or charging it is occur in tRNA, that is known as the charged tRNA. What is the meaning of charging or amino acylation? Excess or addition of a process of addition of amino acid to the tRNA. Can you understood all of you? The process of addition of amino acid to the tRNA molecule. Process of addition of amino acid to the tRNA molecule, that is known as amino acylation. Process of what? Additions of amino acid molecule to the tRNA molecule. Amino acid to the tRNA molecule, that is known as the charged tRNA. That process is known as amino acylation or charging. Without the addition of a tRNA, without the addition of the amino acid to the tRNA, that is known as the uncharged tRNA. Can you understood all of you? In this process of amino acylation, it is essential one of the enzyme. That enzyme is known as amino acyl tRNA synthetase. Okay, amino acyl tRNA synthetase enzyme, it is involving to bind the excess of or addition of amino acid to the tRNA molecule. Okay, if two such tRNAs are brought together, peptide bond formation is favored energetically. When the charged and uncharged tRNA molecules brought together, the peptide bond formation is formed energetically. Okay, so here what amino acyl tRNA synthetase enzymes is present. This enzyme, what is the main uses of this enzyme? It is helpful to add the excess of amino acid to the tRNA molecule. Okay, so that time, in that process, that time what? Peptide bonds, it is when we get the two amino acids are, two tRNA molecules are come together. One is charged tRNA, another one is uncharged tRNA. Okay, so what is the meaning of charged tRNA? Excess of amino acids attached to the tRNA molecule. Can you understood? Excess of amino acid molecule attached to the tRNA molecule, that is known as the charged tRNA. When this charged tRNA and uncharged tRNA brought together, okay, that time it is helpful to favor the formation of a peptide bond. Okay, so that time peptide bond it is favor energetically. They can form formation of the peptide bond very energetically. Okay, energetically peptide bond formation it is occur. Okay, that time, which time? When we are brought together. Okay, two such tRNAs are brought together, peptide bond formation is favored energetically. Number of amino acids are joined by peptide bonds to form a polypeptide chain. Okay, number of amino acids joined to the peptide bond. When the number of amino acid it is joined to the peptide bond, okay, so that time it is helpful to formation of a polypeptide chain. It is producing in a length large number of chains. That polypeptide chain it is forming. This amino acylation is catalyzed by the enzyme amino acyl tRNA synthetase enzyme. With the help of the amino acyl synthetase tRNA, that enzyme, it is helpful to bind the excess of amino acid to the tRNA molecule. Okay, that process is known as the forward charged tRNA. When the excess amino acids it is not bind to the tRNA molecule, that is known as the uncharged tRNA. When these two different kinds of tRNA, what is the two different kinds of tRNA? One is charged tRNA, another one is uncharged tRNA. So the charged and uncharged tRNA brought together. Okay, when it is brought together, that time polypeptide bond formation of a polypeptide chain it is favored energetically. Polypeptide chain they can formed, okay, energetically with the help of the enzyme. Which enzyme? Amino acyl tRNA synthetase enzyme. Okay, so number of amino acids are joined by the peptide bond. இந்த மாதிரி நிறைய amino acid இந்த peptide bond மூலமா என்ன செய்யுது join ஆயிட்டே வரும் அங்க tRNA molecule. அப்ப ஆட்டோமேட்டிக்கா என்ன ஆகும் they can produce the polypeptide chain. அதே peptide bond நிறைய வந்து ஃபார்ம் பண்ணி join ஆயிட்டே இருக்கிறதுனால they can produce the polypeptide chain. Can you understood? This is an endothermic reaction and is associated with ATP hydrolysis. ATP molecule அங்க hydrolysis ஆகி ஆகுது இது break down ஆகுது அதை நம்ம என்னன்னு சொல்றோம் endothermic. So release of the ATP molecule. 20 different amino acid tRNA synthetase are known. Already they known very well 20 different amino acid tRNA synthetase. The power to recognize codon on the mRNA lies in the tRNA molecule and not in the attached amino acid molecule.